is not easy in carbohydrate chemistry to synthesize selectively patterned molecules, and that is something which has posed a challenge for people. Not many people understand the importance of the pattern of acetylation, which I am going to highlight today. And after we understand the importance of it, how to develop that, but only biotechnological approaches can help us to do developing such type of polymers. And all of us know those who have contributed for sequencing of nucleic acid has got a Nobel Prize. So those who have contributed for sequencing of proteins also got a Nobel Prize. And in both the cases, it was Friedrich Sanger who got a Nobel Prize for that. And this is something I was trying to tell you, that there is a degree of polymerization. You realize that is the number of residues that are there, and then the degree of acetylation, or you also express in terms of fraction of acetylation, the relative abundance of these N-acetylglucosamine molecules on the polymer or oligomer is what we refer as degree of acetylation. And the pattern of acetylation is about the sequence of the N-acetylated residues and deacetylated residues. A residue and D residue, we call it AD residues. Now I take you to, because I said there are three things that I need to introduce. One is chitosan, one is immunity, one is the symbiosis. This is a classical symbiosis case where you find the rhizobium responding to a flavonoid molecule secreted by the plant, and in turn produces a lipopolysaccharide molecule the signal molecule from the NOD factor that is coming from, and then there is a response from the host plant. And similar things like a NOD factor, you find a mycorrhizal factor also produced by fungi. And the situation becomes complicated when it is fungi. When you have fungal system, when it is interacting with the plant can generate chitin oligomers, because the fungal cell wall is made up of chitin, and the mycorrhiza is secreting a mycorrhizal factor. And when these two are present, how the plant has to take a decision is a very interesting thing. Now we have to see what kind of informations are there about the lipid chitin oligosaccharides. They are primarily chitin oligosaccharides which have substituted additional lipid chain attached to that. That is something which we need to understand. The entire recognition process between the plant and microorganisms is primarily focused on the chitin-chitosan structures that are originating from the microorganism, be it a symbiotic organism or a pathogenic organism. And you can see the synthesis of lipid chitin oligosaccharides in rhizobium, which requires core genes involved in chitin synthesis, chitin deacetylation and acetylation, and other steps are required for the addition of the functional groups. So everything for a symbiotic organism, also these chitin synthases and chitin deacetylases are responsible on one side, and for the synthesis of chitin also chitin synthases are responsible. This is one area which is still, I would say, a gray area, where we have a lot of importance attached to the lipid molecules and mediated signaling in plants. There is some growing evidence that lipid related molecules, which are most of them are shown there, and all of them are important in signaling between the plant and microbe interactions. There was a wonderful review in Trends in Plant Sciences in 2022 on that. And this kind of signaling processes are being studied throughout the world. Now how the pattern recognition receptors at the cell surface and the nucleotide binding, the leucine-rich repeats in the intracellular, how these two are interacting with the molecules emanating from the microorganisms is being studied. One quick summary of it is there are two types of receptors that you find. One is the receptor kinases. The top panel is talking about the receptor kinases, which have a kinase in an intracellular kinase domain and an extracellular domain, which interacts with the molecules coming from the microorganism. Other type of receptors that you find are receptor like proteins. They are called as RLPs. Varieties of these molecules are known to be present on the surface of the plants, which interact with a variety of molecules that come from microorganisms. And a small summary of it, I am trying to narrow it down. What are the most crucial ones for plant microbe interaction? That is the receptors that contain the receptor-like kinases and receptor-like proteins that have an extracellular lysine modules, 3 tandem modules of lysine when it is present, they are the ones which are important for recognition of the carbohydrate molecules that are coming from microorganisms. This is what I am trying to show. You can see in all the 4 cases, in the first case there is a NOD factor with which it is when it is interacting with the lysine module containing receptor-like kinase, when it is interacting with the NOD factor, it activates the symbiotic pathway, allows the colonization. And similarly, when it is interacting with the MYC factor, it allows colonization. And look at the extreme other side, the left side of it, you can see when it is the chitin molecule which is interacting, which is not the LCO, it is the chitin which is interacting with the lysine modules extracellularly, then you see that there is an MTI, that is what I was talking as a PTI. And the other one is also like a PTI when peptidoglycan, that's another carbohydrate molecule coming from a bacterial pathogen like Pseudomonas. So when pathogen is presenting a molecule, the plant is responding with immunity, and the symbiont is presenting a molecule, both are identical, and similar kind of a receptor is interacting with them. There is a hypothesis which has been put forward, which has solid evidence, solid ground for it. When the molecule coming from the microorganism has a lipid component, the lipid chitin oligosaccharide, then it activates the left hand side, the common symbiotic pathway, CSP we call it, and on the right hand side we see the carbohydrate, only the chitin oligosaccharide, you find more of the pathogen triggered immunity. That means there is something to do with the lipid attachment that is there to the signal molecule that is coming from the microorganism. So that's where I was trying to show the lipid mediated signaling which is gaining importance in plant microbe interactions also. So the molecule as attached to the carbohydrate moiety, and also the molecule without attached to the carbohydrate moiety, lipids are playing a very significant role. Lipid related compounds are playing a significant role in plant microbe interactions. And this is a classical example which was shown in PNAS long time ago, some 10 years ago, which was shown where a chitin octamer, when it is interacting with the receptor, it induces dimerization of the receptor, and also it co-recruits an additional binding chitin ester binding protein. CEBIP is the chitin ester binding protein that has been shown by Shibuya's group from Japan. This is the kind, again, a simplistic picture of that. And now I enter to what we have been doing around this. We are working on a process what we call defined as transglycosylation, and it is when a chitinase enzyme acts on a substrate, it is supposed to cleave the molecule into smaller molecules. Here is a chitinase which we have reported in Journal of Bacteriology long time ago, and subsequently we have done a lot of things around it, and also a couple of patents filed on that. And in this transglycosylation process, this enzyme not only cleaves the longer polymers into shorter oligomers, it can take the shorter oligomers and can synthesize a longer polymer. And synthesizing a longer polymer is always a difficult task in a chemical synthesis process, and we thought this is a wonderful candidate to do that, and that's where we concentrated on this particular enzyme. And we have done a lot of structural analysis of this protein, and initially we have done bioinformatic analysis, subsequently we have even obtained the crystal structure of the protein, and we have carried out several mutations where we could see there is an improvement in the transglycosylation process, whereby we can produce more of these transglycosylated products, more of these oligomers. Remember I was talking about an octamer inducing dimerization. That is, longer oligomers are important in dimerizing or interacting with the receptor, so our interest was to produce longer oligomers. And this is the structure, and we recognize that there is an important loop structure which is between the upper half and lower half of the protein, which is very crucial for the biological activity of this particular protein. And we also did some other studies where the substrate entry and substrate exit of the protein has been mapped. And our assumption was, if we close the exit site by introducing a bulky amino acid residue, if we can retain the enzyme substrate complex for a longer period, allow interaction, more of a transglycosylation products, and it indeed turned out to be the same. And when we did it, we could see that there is a significant improvement when we blocked the exit of the products from the protein for a longer duration, there was increase in transglycosylation. We also did some other alternative approaches where we have fused the different domains from different sources. Using some bioinformatic approach, we identified those domains and brought in, and we tried to further improve the transglycosylation process of that. Then what did we do with that? And we have been, by then we were already collaborating with a German group at University of Münster, Professor Bruno Moschbacher's group, and who were also interested in working on, in fact, they were the major group working on structure-function relationship of these chitosanes. So we also joined, because of our experience with the transglycosylation, we have joined that consortium of European consortium, which consisted of about 21 partners across the Europe and India. And a lot of work we have done on generating these oligomers from differently acetylated chitosanes and analyzing those products and looking at those products and their capability to prime the plants and induce immunity in the plants. That is the approach that we have followed. And the two major contributions, first thing was from our own lab. That was in 2015 we have published. And after that, we have, as I said, in our collaborative effort with Bruno Moschbacher, that one was published in 2020, where it involved more work, and we were collaborators with that, and we are partners of that particular publication of Journal of American Chemical Society as well. And this is how we analyze those oligomers that are generated after treatment of these chitosanes. We had to start with very well characterized, differently acetylated chitosanes, and we start with that, and then we start analyzing these products. And after treatment, and we do with this size extrusion chromatography, we separate them and then try to test them. Each of these fractions 12 to 17 generated by this, and with the wild type strain, and with the chitosane which has 61% deacetylation, they were analyzed through multi-top analysis, all that. Finally, we have narrowed down to DP8, degree of polymerization. Chitosanes with degree of polymerization 8, with 8 polymerization 9, DP10. And purified oligomeric fractions of showed a very significant increase in the activities of the enzymes related to immunity in the plant. And since we know O. Conrad is a very well-known researcher in priming, plant priming, and we have looked at these molecules as agents for priming. And our assumption was, jointly, our assumption was that not only the degree of polymerization I emphasized and degree of acetylation, it is the pattern of acetylation on these oligomers also it could be important in inducing the biological activity. And in our case, it is the induction of the priming system. That is what we try to understand. And as you can see here, the monoacetylated residues on the of DP4 have shown excellent priming activity, whereas the monoacetylated residues on the extreme end and the completely deacetylated or completely acetylated residues do not have any biological activity. And taking clue from there, and we try to synthesize, making use of different kind of chitin deacetylases, we try to generate the oligomers of defined pattern. The deacetylated residue, you can see 4 different types of deacetylated residues that we have, a tetramer with defined pattern of acetylation we have generated, making use of these residues, and that we could see. Now you see the one which has at the non-reducing end, the acetylated residue has the highest priming activity, whereas the other ones which at the reducing end has very less of activity, which clearly demonstrates that the pattern of acetylation on the molecule is also very important in inducing biological activity. That's what we have shown in this particular paper. And this is a continuation of the same thing for the results, that part of it I have shown. And what we have also done is we obtained, Sharma is sitting in this audience, is my student. He has developed a process making use of those improved enzymes. a transglycosylation process, set up a transglycosylation process and separated out the oligomers prepared from that and made use of these oligomers and tried to test them in two different ways, as root dip treatment and or as a foliar spray. And foliar spray treatment induced elicitation response early and sustained for a longer period. This was also published a couple of years ago in carbohydrate research. I will skip these publications part. Yes, the conclusion of it, I would like to show that the chitin and chitosan fragments, the DP is important, DA is important, and PA is important. And if we can define and produce, identify, define and produce the molecules, we are going to have the third generation chitosans, which are going to, I will tell you what I mean by third generation chitosans. I just wanted to again bring back the original concept that I introduced. A combination of these chito-oligosaccharide and lipo-chito-oligosaccharide recognition promotes arbuscular mycorrhizal association. When both of them are there, still there is a symbiotic pathway. How is that happening? LCOs, when present in a mixture of along with the chitin oligosaccharides, drive a symbiotic outcome, and this mixture of signals is essential for arbuscular mycorrhizal establishment, has been proven in one of the recent publications that has come in Nature Communications. That was not our work. I am trying to tell you that this is what is where the research is going on, and the cartoon form of that is a model of the chitin fragments and LCO recognition during symbiosis. That was these were the authors Feng et al. In the Nature Communication. This is my collaborator's article. Biotechnologically produced chitosans with non-random acetylation patterns, that means defined patterns, differ from conventional chitosans in properties and activities. This is another thing that they have published. We are not co-authors in this particular work, but this is another work that they have published. This means there is a code, for the chitin, and to some of us it may look like I'm talking about what I'm talking is a fiction, but it may be a reality tomorrow. A lot of things we have, there is a reason for us to believe that these things are happening, and there is a way forward for us to see how best we can produce these kind of molecules, for us to take advantage of these recognition process of the plants, how the plants recognize these microorganisms and how to make use of that. This is what I was talking about. When people were not knowing much about these chitosans, the chitosan was applied. What they were using, about 40 kilograms per hectare, was used for crop protection. And second generation chitosans, where a little of deacetylation things were known and degree of polymerization was known, only about 4 grams per hectare was required. That's what the today's water soluble chitosans that are available plenty in market are coming. And what we are talking about is the third generation chitosans, which are defined degree of polymerization, degree of acetylation, and defined pattern of acetylation. And what we require will be a few micrograms of that. When we have these products available in the market. And I would like to thank all my colleagues, my students who have been with me in this journey. When we have started sometime in 2005, and we have ended up here as of now. And I would like to thank my collaborator Bruno Moschbacher, and also I would like to thank the IPS Executive Committee and particularly Professor Chandra Naik for bringing me here to share our experience of working with chitosans. Thank you very much. I hope I stuck to the time. Okay, thank you.
I do have a question.
Yes.
How long do these immune responses are maintained? So once you treat the plant at the beginning, is the immune response maintained throughout the season, or do you have to provide multiple treatments?
Multiple treatments are required. What we notice is it stays at up to about 3 weeks.
I had dabbled with chitosan on Fusarium wilt of Salaria. That was in 1996 or so. So it was not a very successful experiment. That's all it was. Thank you.
Sir.
Yes.
Sir, now there is so many places people are working on chitosan. and it's almost now it is a area of nanochitosan. Many people are trying to use this to induce signal as an agent. And how far it will be successful coming days?
See, the source that they are using for nanochitosan is very important for us to really understand what it is going to do. In fact, we have worked with chitosan nanoparticles. We have made use of the chitosan as a carrier, and we have entrapped the herpin, a bacterial elicitor molecule, into the chitosan, and on that we have a couple of patents already, and which we are also negotiating. In fact, we are when I was sitting in the morning in the audience this morning, final sanction letter from DST has come, where we are now working with an industry where this chitosan nanoparticle based approach to induce immunity in plant is being has to be started. The final sanction letter has come along with industry. What I meant to say is, unless we know the source and the kind of chitosan with which we are working, it is not going to be easy to tell. We will get some effect. The effect what we get may not be the best effect that we are looking for.
Thank you.
Interaction. And I request next keynote speaker is Dr. Krishna Reddy M. I request Dr. R. Vishwanathan sir to introduce our keynote speaker now.
So as we know, next speaker is our co-chair, Dr. M. Krishna Reddy, and he is one of the pioneers in plant virology in the country. And he had his education from different states, and finally he had his PhD from IRI under the guidance of Professor Anupam Vermaji. And also almost he has worked extensively and throughout his career at IHR in various capacities. And recently he is retired from the service, but he will continue there as an emeritus scientist. And to his credit, he has been recognized by various organizations, and including postdoctoral fellowship, and his area of expertise is on especially on plant virology, molecular plant pathology and biotechnology, host-path interaction, and molecular diagnostics, virus resistance, and related areas. And he has been heading the important project, the network project on plant virus diagnostics. And that almost he was heading the project where the project was located in different centers, and he has ably guided, and 2 EFCs he has guided, and also he has supported the plant virology work across the country. And now I invite Dr. Reddy to present his paper.
Thank you, Chairman. At the outset, I thank the organizers and the executive members of IPS. It is indeed a privilege to be in IPS and present a keynote address. And my topic is about the integrated management of virus diseases, especially in vegetable crops. And if you see, the vegetables are very important in the world. China ranks first, India is the second, and totally 73% of vegetable production takes place from China and India. And China and India contribute to the even vegetable seed production, which is the world beating condition. The intensive cultivation and extensive cultivation of vegetables has several problems, out of which the biotic stresses, viruses are the major. And you are all aware that currently the viruses are taking significant losses in vegetable crops. And in addition to the climate change, which is influencing the insect vectors, which are the primary responsible for virus spread, and the trade in globalization of horticulture crops, especially vegetable seed and planting material, is also responsible for heavy emergence of vegetable viruses. Of the 15% of global production, if you see, plant diseases are the one responsible. Among them, the major one is the 1/3 is the plant viruses. Though it affects the cultivation as well as yield and in terms of loss and the quality, and marketability. Often the farmers and also we recommend the pesticides to curtail the virus diseases only, to stop the insect vectors but not the virus. Till today we don't have any chemical which can directly affect the virus. As you see, the number of virus genus which are present at least in the intensive vegetable cultivation in Asian region, more than 32 genera are there, of which there are several viruses. All of them are not economically important. If you see the major groups of viruses which are divided, the ones which are transmitted by aphids, and the ones which are transmitted by whitefly, and the ones which are transmitted with thrips are the major ones, and some extent the nematode ones were very in less number. But there is a group of viruses which are transmitted through seed and as well as insect vector, the central one. And if you see the number one group, whitefly transmitted, there are several bigomoviruses. The bigoma is a single stranded DNA, which is the whitefly mediated. It is in several vegetables a serious concern. And again another whitefly transmitter is the crinoviruses. This group of viruses are transmitted by two species of Bemisia, Triolidis and Bemisia. Both, one is glassos, another is the field whitefly. Both are capable of transmitting these viruses. And in thrips cases, the orthotaspoviruses are the ones which are major. Among the Asian countries, groundnut Burnacrasis, Capsicum chlorasis and tomato spotted are the ones which are a serious concern in several vegetable crops. And among the aphid transmitted viruses, cucumber mosaic virus, earlier known to be less important in solanaceous vegetables, currently in Asia and also in some parts of Europe, the virus is becoming serious and its behavior is changed. Earlier it used to express in the plant the symptoms what you see in the top, but currently it is expressing in the fruits but not in the plant, and then leading to the total market yield loss. The another group of aphid transmitter are the potyviruses, which again is very serious concern in solanaceous and cucurbit crops. And recently the aphid-borne polariviruses, which are persistently transmitted, are again a serious concern, and often the symptoms are confused with the whitefly transmitted diseases. And there are few viruses which are mechanically transmitted or externally seed transmitted. Those are the three viruses which are earlier known: tobacco mosaic, tomato mosaic and pepper mild mottle. But the two newly evolved viruses for last 2 years is a very concern throughout the world, especially quarantine purpose, and there is a serious restriction on the movement of these viruses because of their seed-borne nature. The viruses, if you see in terms of effect of the climate changes we experience, the climate change not only affects the virus incidence, increase in virulence and host range, and also the vectors which are the major ones will lead to the increased temperature increase, make the susceptibility to the insect vectors, in turn leading to new viruses, new strains and spread in the newer location in extreme conditions. As a virus, if you understand the initial virus, the infected plant, from their transmission through insect vectors lead to the healthy plant either through pollen or through seed, and then again through insect, and the cycle continues. So here is the one where we need to emphasize. Often people will say that in virus diseases there is no control, it's only cut and burn or throw it. It is not like that. It is the nature of understanding the pathogen very important. And in this case, what we need to understand is the prevention, first step, and then the decision tools, and then solutions how to implement them for managing these viruses. As aware of as any plant pathologist, we know that host plant, the interaction, disease development in the host pathogen interaction, the three triangle. Whereas in case of viruses, it is the one which is the fourth factor, is the insect vector and or the tripartite interaction, virus, vector and host. Then environment influences all the three factors. The fourth factor, which is very critical for successful development of the virus disease, and therefore any attempts made for the managing we need to understand and then intervene in each step how to prevent this disease development. As a first step, we to take in case of viruses, the first stage is the production aspects. In the nursery stage, seedlings should be grown under nylon net at the 40 to 60 mesh, because most of them are transmitted by the sucking pests, the aphid and several sucking pests, which are very minute. So the nylon net used, often people as a misconception, they use the mosquito net, which is a higher mesh, which is not useful, but the critical is the one which has 40 to 60 mesh. And then the early incidence of disease eradication will also reduce the subsequent spread in the crop cycle. And the agronomic like non-host barriers, especially cereal crops or tall growing crops growing border, which can reduce at least the non-persistently transmitted aphid transmitted viruses. We have done extensive trials and adopted for many farmers that seedling production. Nowadays, the seedling produced under the nursery conditions, most of the nurseries are not scientific. They use the shade net for the multiplication, which is not a insect proof net. Whereas any nursery practice should be under nylon net, which is 40 to 60 mesh, which are scientifically produced. And if you see the seedlings, the quality in terms of the virus presence is nil in the beginning, so that the seedlings are protected in the early stage and will be having healthy plants in the ground. Further, to act develop the resistance or the strength in the seedlings, we need to apply bioagents, biopriming in the seedling stage, so that they go very effectively. And in terms of management, the manipulation of host and the arthropod vectors is a very important area. Now current research is change in the orientation and behavior of insect vectors to attract and repel the host plants. And there are several research carried, and we also have done some external work on this area, where the volatiles emitting from the plants are the responsible either attraction or repellence of the vectors, and that leads to the very efficiency. And there are ample evidence now, the different groups of viruses, if you see, the polariviruses, they affect the growth rate, alate production, dispersal and fitness of the aphid vectors, which in turn, if you see this case in the case number 1, wherein the infected, the healthy plant when infected, certain volatiles are generated, and these volatiles are recognized by insects vectors, and they in turn feed on them, and then once they acquire the virus, again virus will modify this behavior of the aphids, will attract to the volatile produced by healthy plant, thereby it is going to feed on healthy plant but not on already infected plant. Once it acquires the virus, it doesn't go again to the infected plant. That is the mechanism how it is there. This is the how now current area of research, the identification of volatiles and change the behavior of vector. Already the using next generation sequence and then the metatranscriptome analysis done with these 3 groups of insect vectors. There are several defense regulated genes which are upregulated and are downregulated when they feed, and these mechanisms again depending on the host phenology, host volatile and genes which they are expressing under biotic stress conditions when the insect vectors feed or the virus enters into the host. The major things which are now happening in case of viruses is the one salicylic acid, jasmonic acid and ethylene pathway. All these 3 are the responsible for activation or suppression or reducing the virus replication. One of the now earlier chemicals were used is the some BABA, the brassinosteroids, silicon and BTH. BTH is the commercial formulation worldwide used except in India. But now Professor Podili has explained the chitosan to you, sir. Now currently chitosan is now has been used inducing against virus resistance in combination with other metal ions as well as with other stimulants of bioagents. So thereby it is leading to the expression, especially there are 2-way interaction which happens in the biostimulants. One is the repellence of the insect vectors, which are the primary spreader of the virus, and the second is in turn virus reduction is happening in terms of replication and movement of the virus in the host plants. And these are the few examples I am quoting, showing that there are several reports which are already published in vegetable crops. And if you see the recent publication where bacillus has been shown to affect in the tobacco mosaic in tomato, wherein 3 genes of polybiosynthetic pathways and 3 genes of PR proteins are upregulated in treated ones as compared to the control, and there is significant defense regulation in these which are reducing the. In our case, we have done in chili wherein 3 viruses, the chili leaf curl, cucumber mosaic and chili vinyl mottle virus, when we applied salicylic acid and aminoliquefluence bacteria, and then a combination of microbial format, that is ANC. In all the cases, for up to 7 days, there is significant reduction in the virus replication or multiplication, but 15th day there is a elevation of this. Though it comparatively it is control, it is 50% less. There is so this makes the determination of the critical interval time, which time to spray, so that they can be subsequently can prolong the host defense mechanism. This is the mechanisms which I am highlighting. Then next we come to the resistance build-up. We have done resistance breeding for tomato leaf curl, wherein we have 4 hybrids developed based on this multiple disease resistance. It is not only resistant to tomato leaf curl virus, but also Phytopathological and early blight and bacterial wilt. And we have a chili again, 4 hybrids developed which are resistant to cucumber mosaic and the chili with both the CME and potivirus. And we have recently developed 6 hybrids for chili leaf curl, which is a devastating for past 2 years throughout the country. And these hybrids we have now currently has undergone the field performance, and it has been demonstrated in 3 different states, and the farmers are very happy. In spite of the black thrip incidents, our hybrids are performed much better and able to have less incidence of chili leaf curl and higher yield returns in terms of yield and quality. And currently, though I am not dealing much on transgenic, the practically transgenic also been coat protein mediated has been developed, and it a few cucurbit viruses, especially papaya ring spot virus and cucumber mosaic virus and Jukni yellow mosaic virus have been done in the Europe and America, but in Indian conditions no transgenic vegetables for against virus are available or not in the practice. Commercially till now the virus resistant chili transgenic lines are not available as on today. And current research is going on genome editing. We also initiated on chili and tomato for potiviruses based on CRISPR-Cas, wherein the elongation factor which is responsible for binding the VP gene, and that we are modifying so that it can reduce the virus binding. This will ultimately the less virus incidence. Some of the examples already known for the genome editing for virus resistance are these are the tomato and in the cucumber and cassava and potato. These are already in the plants are ready, but till commercial cultivation these varieties are not yet come. And when you come to the resistance part, and then the overall management is the vectors which are very critical for these viruses. The control depends on the interaction of this all the 3 types of behavior: non-persistent, persistent and semi-persistent. Spraying is the most commonly used method, but it is not useful for non-persistent transmitted virus, because the moment you apply the insecticide, the virus is already inserted and the symptoms will develop. Then the next option is the preventing the landing and changing the behavior. That is the water we use, the reflective mulches or barrier crops or oils to spray, so that they deter or the penetration and feeding behavior of the insect vector are changed. And some of these trials what we have tried in case of chili, tomato and capsicum, wherein the growing border crops with tall growing maize, then followed by silver mulching, then use of yellow and blue sticky traps, then application of selective pesticides in the initial up to flowering period only. No pesticide is recommended after flowering or fruit set, because the detention and the residual problem. And not only that, the incidence of viruses once the fruit set or the economic part is developed is less than 5%. The yield loss is not there, so there is no need of spraying these chemicals. And in addition, farmer do not keep quiet because he has a tendency to use insecticides continuously. What we advise them to change their psychology, application of micronutrients and the seaweed extract or the microbial formulation, which in turn strengthen the biostimulation of the host, strengthening the immune system of the host, so that will take care of the production yield level, and there is incidence, even it occurs, it is less, not causing much yield loss. And if you see some of the trials what we have done, if you see the water cropping, mulching, you can see the excellent yield obtained, 3 to 4 times higher than the normal ones. And it is same thing in case of Chilean capsicum. The farmers are adopting in Karnataka and also Tamil Nadu and even in Maharashtra. And we also done in case of watermelon against watermelon viruses, and cucumber and muskmelon, and in bitter gourd. Now bitter gourd is seriously affected by the whitefly transmitted viruses and the polariviruses, which are very serious concern. We have successfully shown, and if you see in our experiments for past 3 years, the application of aminoliquins and the seaweed extract and neem seed oil has contributed higher in the production of yield, whereas the chemicals, acephate and fipronil insecticides which are used are not on par with this one. It is higher, these things. It is merely not that virus is not there, but virus incidence is reduced and the effect of virus is not seen much in these treatments as compared to the insecticides, whereas the ultimate the aim for the farmer is the economic yield. If yield enhancement is there, even less incidence of virus doesn't make the loss. And the future of this is the emerging plant disease management depends on the progress developments in terms of disease surveillance and identifying proper cause and the spread of the disease, and then improved disease detection systems to use proper detection of viruses, and communication and giving forewarning systems so that we can develop early information farmers how to take preventive measures, and predict the plant disease modeling and decision support systems to develop in different agro ecosystems how to use these management technologies, and integration of all these technologies is required. No single technology or method can stop the virus disease. It is the integration of all methods from seed sowing to the final harvesting. That is therefore we can definitely curtail the viruses, and it is also too important. We need to integrate other systems of science so that we can bring effective management of viruses in future. And these are my collaborators and my colleagues who have worked with us, and thank you one and all.
Thank you, sir, for completing your lecture in time. And in a nutshell, with all your experience, you are able to come up with a combo package for managing the viral diseases, especially in vegetable crops. So for either north or south where we face this problem, so I think your guidance is still required to manage these emerging viruses in vegetable crops. You can take 1 or 2 questions. Yeah. Yes, sir.
Thank you, brother. I would like to congratulate you because first time I had heard such a wonderful lecture by virologists beyond the diagnostics and genomics. So practical utility, lot of attention you have given to the audience. This is congratulations to you. As I have seen personally, the good agriculture practices and insect control, if you can, vector control, if you can do, we can escape the viral disease. That is good. And you have mentioned very rightly. Elicitors, chemical elicitors, as already has pointed out earlier, this is not providing adequate protection for longer duration. We have seen, especially in fungal disease cases also, maximum 2 weeks. So what in the case of whether this we can recommend to the farmers use this, or this has to be integrated with biocontrolling agents or some things like that. especially for the virus resistance breeding. Personally, this is also my personal observations. I may be wrong. The most of the resistant varieties not durable for longer durations as compared to other fungal and bacterial diseases. So whether we are not focusing to the vector and virus virulence together, maybe our program breeding program. Why I am asking, because maybe we have tuned our the program at the national level, whether the tuning is required to focus the resistant breeding, considering the vector, not only the virus alone, or such a frequent mapping of the virus population based on the host differential. We don't have information, because in IRI working, I have the experience. 2 genome sequence, nucleotide sequence, new virus, Galai virus, Assam virus, Andhra Pradesh virus. Nobody has testing them on the set of differentials which can be used this information for the breeding program. We are liking in this direction. And what is your opinion?
You have asked, sir, all questions in one this thing, and my answer is entire topic. Sir, the first question what you mentioned is the disease management, the bioagents. If you have seen, I have mentioned the application timing of the stimulants and bioagents, soil, and also at 30 days and 45 days or 60 days, only 3 space, because initially we are recommending insecticides. I mentioned clearly up to flowering period we need to protect insects. Later on, insects, even if it comes, the virus comes, the effect on yield is reduced drastically. But our psychology is the single plant is shown symptom, farmers will not keep quiet, they will spray every alternate day, which is leading problem for us in terms of resistance build-up in the insect vectors, as well as poison, contamination and environment and the accumulation in the food chain. So that is why our recommendation is insecticides to be sprayed before flowering. The economic part develops later, so residues are not built in the fruit formation. That is why biostimulants we are recommending after the stop of insecticides. Gives satisfaction for the farmers that he is spraying something. At the same time, these are not harmful and will build the innate immunity trigger, so that it is not full protection. It is some difference. Once the fruit formation is developed, there is nothing is required. What we say, economic part is developed, disease comes also, it doesn't make loss. That is the concept behind that. The third one, what you asked, resistance point of view. If you say resistant breeding as compared to fungal disease, virus is also stable resistance is there. But our way of defining resistance and our stable resistance is isolated or not. Often in a hurry or to jump into quick, we release a partial resistance and claim as a resistance which breaks easily. That is one reason. The second one is, in case of potty viruses and cucumber, we have varieties which we have developed 10 years back, still our chili varieties are stable, resistance is there. Whereas your question is appropriate for bigomoviruses, single stranded DNA viruses. The recombination development of virus is very fast, resistance breaking pattern is very fast. So for this, even if you develop a variety for 3 years is excellent, I rate it as an excellent. But breeding is not a one-time stop point. It is a continuous process. As change in the strain evolves, we need to bring another line for that. So there is definitely we can increase the resistant pattern. Broad genetic base identification is the critical issue. As you pointed even the other day in the diagnostics, whether is it work in this. That is where critical areas are. We don't identify huge gene pool available in the wild species. Unless we bring more number of genes, then only we can get a stable resistance, which can we claim that yes, my variety can perform. That is the slow process which is lacking. We need to hasten up this, bringing more pre-breeding lines, bringing more genes from wild, so that we can get a stable resistance source for this. I think I answered.
Thank you, sir. We will take up the next.
Thank you, sir, for your informative lecture.
T.S. Venkatraman Award and Puttarudraya Memorial Endowment Award, Syed Ahmad Khan Memorial Award, and he has been also a founder and shown the evidence that in sugarcane, we all know sugarcane yields in India, whatever we are getting, it is the excellent yield. But it is the first time demonstrated, all of them have virus. Without virus, there is a 20 to 40% enhancement of sugar yields when virus is not there. In this direction, he has worked the production of virus-free planting material and generating through tissue culture and supplying to sugar industry, and contributed vast in the sugar improvement program for past 5 to 10 years. I now invite Dr. Vishwanathan to share his knowledge for the audience.
Thank you. Thank you, sir, for this lecture, this August gathering. So I will be presenting on most on pathogen variation in a red rot, that is important pathogen. So of course I don't touch on sugarcane viruses in this lecture. So coming to sugarcane and red rot, so the both at least the host and also pathogen cannot be separated. So that is either in the form in the field or in the research front. So that's the importance for we give to the disease. Since when the disease comes, it's almost causes 100% losses, up to 100% losses is possible. So that's what that we have been working on the disease for more than 100 years in the country. But still, of course, where we started, still we are at the same place. In the sense we have red rot epidemics, severe epidemics that we will be seeing. And just one line on the crop scenario. And we know that now sugarcane India has been in a unique position. So that is India is occupying the number one position in sugar production. And of course, the area wise, Brazil still tops in the world. And of course, they divert most of their sugarcane for biofuel, bioethanol production. And coming to Indian scenario, we also now India started exporting sugar. For the first time, India has occupied second place in sugar export and earning about 40,000 crores of foreign exchange. So that is a very big achievement in this sector. And apart from that, also India has at least the green energy program, and already we have achieved 10% blending of bioethanol in petrol. So though the target was fixed much later, but we were able to achieve the 10% blending, and we have to achieve or to reach 20% blending in another 2 years. Probably with all crop production and protection technologies, we will be able to achieve this target. Though it is a big target, so we will be able to do that. And in the present lecture, so my lecture will devote more on the red rot epidemics and also on the pathogenic variation and what was the work which we have done on omics, that is genomics and the transcriptomic part of this pathogen, and also to for some aspects on functional validation of the pathogenicity genes. So as I said earlier, this both sugarcane and red rot are almost were together for so many years. The reason is that, and we have found that many occasions, we found the breakdown of resistance due to evolution of new pathotypes. So that's how we are continuously evolving new varieties. And also, but another important thing, that pathogen variation is also happening. But only way so far we are able to manage the disease also through varieties. So that's how we have released n number of varieties to manage the disease under field condition. And that's how that now even today a variety can be released for commercial cultivation if it possesses high yield, high quality, that is high sucrose and red rot resistant. So still that we see maintain that very rigid strategy to manage the disease. And just if you see this overall, that last several decades, we had the red rot epidemics across the decades, but almost many elite varieties we have lost. And currently also this our variety, that is CO0238, probably you must be remembering a few days before Dr. Bachiram was and honored with Padma Shri for his work on evolving this particular variety 0238. This particular variety is even today is occupying more than 80% the subtropical region. That is, it is almost occupying more than 40% of the cane area in the country. So that is how we made a big revolution, but also we have found there is a severe red rot also in this field. And just taking you back and how this really pathogen variation, how sugarcane pathologists identify the variation in the field, and it is something like evolution of light isolates. So this is how that when you people who have worked on Coltotrichum and related genera may be knowing that light isolates are and also dark isolates. In this case, we found, and mostly this when we see a severe epidemics means there will be evolution of light isolates. So that's how we see that highly sporulating like isolates, which that's how we say now we say that also hypervirulent isolates. When you have a collection of isolates, always you will see these isolates, some are on the top are highly sporulating, whereas in the bottom are almost a matte type of one. So they are also pathogenic, but they are less pathogenic. So this is how we find variation. So then these are all the petri dishes we see these cultures. But to assess the pathogenicity, we wanted to have developed or identify a set of differentials. So that's how over the years we worked at the Sugarcane Breeding Institute, and we identified a set of differentials, and then we were able to designate the pathotypes then and there. Another important thing, and when since it is a vegetatively propagated crop, and we found this different varieties and also even pathogenic variation also we found in the same variety. So we could see several isolates. That too, I could get more than 150 isolates. But the testing pathogenicity in this crop is such a tedious one under field conditions, where we have tested at 117 isolates and then grouped them. Then finally we found still we are unable to group them. Both we conducted Karnal and Coimbatore, that is representing subtropical and tropical locations. Still we could see that we could not categorize the isolates whether it's tropical or subtropical. But what we found, this variation was mostly attributed to the host variety and also place of collection, and to some extent on the host the location of testing. So what we find in this particular picture, where many of the isolates I have collected particularly from Tamil Nadu, and same variety exhibits so much of variation for the pathotype, and there we adopt a method called plug method of inoculation. So we introduce the pathogen at the bottom of the cane, and then we assess the linear development of the lesion. So that's how you can see such a variation we find. And also coming to another important aspects of identifying the new pathotype, how the pathotype emerges under field conditions. Really, if you see this particular case where we have identified a new pathogenic race, that is we normally designate the pathotype, that is CF12, that means Coltotrichum falcatum 12, and where that you can see that one it is compared against the previously established one, that is CF06. That is the one which has been responsible for breakdown of resistance in the popular variety COC 671. So that was the ruling variety in 80s and 90s. So that's how the entire tropical region, this variety went out of cultivation due to red rot epidemics. So what we found that the new pathotype has almost comparatively higher virulence. Even among the for several years, 7 years of testing also you can see. Overall you can see this new pathotype has almost in the last column you see more number of susceptible reaction, indicating that the pathogen has slowly adapted and it is able to cause susceptible reaction on the many of the resistant hosts. So we tested with a set of varieties. So that's how we can find, even in a set of differentials also we could see, and this particularly the new pathotype is able to establish many, and we can see susceptible reactions. So this is how we found that new pathotype has emerged, and that's how even of course it's also the other pathotypes also were collected, but they also behaved similar to the new pathotype. And this is how that finally what the conclusion on this after identifying the pathotype was. So that when we remove the variety from cultivation, though this particular pathotype from that variety maintains virulence, but it is a moderate virulence, whereas the new variety when it succumbs to the pathogen, so where the pathogen evolves quickly and gets or even acquire more virulence. So that's we suggest we found there is a similar to post adaptation we found in sugarcane. And coming to these particular important aspects on the current scenario and the boom and bust cycle. So as we see in the past several times we found here this was reported based on the examples of Bihar, where the several cycles of boom and bust cycle was found in sugar production that was correlated with the sugar red rot epidemics. So that's how then they have defined that it is closer to or analogous to vertifoliation effect. It was reported in potato, and a similar thing is currently we are facing the issue of boom and bust cycle. As I was mentioning with the 0238, this has almost has been adopted in more than 90% of the cane area in the subtropical region. So that has recently it has broke down. So that's how this was almost newspaper reports. And that it's so much almost monoculture which we have